Welcome. I'm Dr. Vinay Prasad. I'm a hematologist-oncologist, and I'm Associate Professor of Medicine at the University of California, San Francisco. In my professional life, I see patients, I teach trainees, and I do research in healthcare policy. This is Plenary Session. Plenary Session is a podcast at the intersection of medicine, oncology, and health policy, and you're listening to Season 3. On this week's episode. This week on Plenary Session, we have a lot in store for you. It's going to be a great episode. I'm going to kick things off with a discussion of whether or not healthy people should be allowed access to investigational vaccines as Phase 3 clinical trials are ongoing. This is an idea that was first floated on the internet, where all great original ideas are first floated. Next, I have a recap on the Adora Study. This is the study that there have been many industry-sponsored debates about, conceding that it is a great study. So I'm going to take another crack at explaining the problems of Adora. Then I'm joined via Zoom by Audrey Tran for questions from a medical student. You won't want to miss this far-reaching discussion. And last, we have the news segment, Journal Club with a Fellow. I'm joined by Dr. Karine Tawagi, and she's going to take us through an article, atezolizumab and bevacizumab in hepatocellular carcinoma against sorafenib. And you won't want to miss this discussion. This is the Imbrave 150 discussion. So stay tuned. And a quick note, I promised that I would talk about a precision oncology study, but this episode got too full, so I got to talk about that next week. So stay tuned for that. If you like this podcast and want more content, follow me on Twitter at vprasadmdmph. Check out the YouTube channel, Vinay Prasad, MD-MPH. Patreon backers will get access to the slides for lectures I give on Plenary Session. Want to hear from us? Email us your question at plenarysessionpodcast@gmail.com. I really don't understand what's going on Twitter and COVID-19. The last few days have seen a flurry of interest around a terrible idea, and that terrible idea is, while the Phase 3 randomized control trial for Moderna's vaccine goes on, and ostensibly several other candidate vaccines which are nearing the starting point of a Phase 3 study, while those Phase 3 studies go on, we ought to allow people who are interested in receiving the vaccine that's passed Phase 1 testing but has not yet proven safety and efficacy in Phase 3 testing in an off-protocol use. And the people who have promoted this have used confusing language like expanded access or compassionate use, language that I don't think they fully understand the regulatory implications of. But let's put that aside. Let's just say their proposal is simple. While the randomized trial is ongoing, we ought to permit other people to gain access to the vaccine. Well, that's a hot take put out in a blog post on Forbes and then on Twitter, amplified and mega amplified because, people who don't know anything about vaccine or drug approval would be quick to think that, boy, wouldn't I want to get a shot? Wouldn't that be lovely? And they're eager to amplify it. But it's a very misguided thing to amplify because there's a million problems with this. So first, let me just talk about what I think the key problems are with this proposal. One, as candidate vaccines are manufactured and built up in increased supply, the most prudent thing to do with that limited supply, that supply you have in hand, is to administer it as rapidly as possible in a randomized fashion. When you want to know whether or not something works, there are a few factors that affect the speed with which you get that result. One factor is the event rate. How widespread is COVID transmission occurring? And if you go into places that are hotter than places that are cooler, your vaccine trial is going to give you a result faster. That's one. Two, the effect size of the vaccine. The bigger the effect size, the faster you're going to get a result. A vaccine that absolutely eliminates all cases of COVID is going to have a signal that's shown rather rapidly. A vaccine with a 5% reduction, it's going to take a hell of a long time. So the efficacy of the vaccine, that's another factor that goes into it. And then the third factor is the rate with which you enroll people in the randomized trial, the enrollment rate. These are the three key factors in time to result in a Phase 3 randomized controlled trial. One thing that I don't think these people fully understand is vaccines are not safe after a Phase 1 study. Vaccines are safe after a phase 4 study, which is a population-based surveillance study, that even vaccines that have passed phase 3 testing have been debuted on the market with intolerable phase 4 safety signals and have been revoked from the market. Vaccines are not safe with the sample size of a phase 1 study. Phase 1 studies do not prove safety. They prove MTD. They prove that the drug or product is not overwhelmingly toxic. That's all they prove. Safety is only understood in the context of efficacy, and safe things are where the net harms do not outweigh the net benefits. Safety and efficacy can only be understood together. There is no perfectly safe compound, unless you're talking about maybe homeopathy, but of course that's also going to be perfectly useless too. Okay, so that's safety and efficacy that these people don't understand. If you're making vaccine as it's being made, the absolute most prudent thing to do is to prioritize it for randomized distribution in hotspots, measuring the endpoint, which will give you the fastest time, the fastest result. And then the moment you know that it's positive, you can put every ounce of energy into cranking up that vaccine production. There are several candidate vaccines. There's no assurance that any of them are going to be positive. Some might be positive, others might be negative. We don't know. So if you allow this proposal where people are getting off protocol vaccine, one, you're hampering the randomized trial because you're diverting the supply of the vaccine. Two, you're hampering the randomized trial because you're poisoning the equipoise that exists in the community. Some people who don't understand medical science, if given a choice between getting a sure thing vaccine off protocol or being randomized in a study, they're going to choose getting the sure thing vaccine and not enroll in the study. So you're poisoning the randomized control trial, which is essential to understand if the vaccine has a net benefit effects. The next thing is, what if the vaccine you give out is actually ineffective? You may have a large number of people. This is sort of in the hypothetical world that we're just cranking out the supply and we have tons of extra supply beyond our randomized capabilities. And you start giving that out. People who get that potentially ineffective vaccine are going to feel as if they got a vaccine, and they may change their behavior, which may in fact worsen a pandemic. That's a possibility. The other possibility is the one that they're getting off protocol might be the one that turns out to be ineffective, and thus later you're going to have to give them a different one, but their appetite for getting a different one might be reduced. The other possibility is that there's some antibody enhancement. There's some deleterious effect of the vaccine, and you actually worsen the situation. You don't know. That's why we do clinical studies. We don't know. It's possible that the unpredictable may occur. Another possibility, the vaccine is either doesn't work or works with a 20% reduction. It's actually a worthwhile vaccine. Maybe 20% is good in certain settings. I mean, it's better than 0% efficacy. You give it out widely, and then 3 people in whom it was received off protocol have Guillain-Barré. Well, is that due to the vaccine? Is it not due to the vaccine? Is it the baseline level of Guillain-Barré, or is it a higher level? What if one of them has ADEM? What if one of them gets really sick with some other malady? And in fact, this has occurred. Osama Bilal has a nice link that he tweeted, which was about a prior pandemic flu season where a vaccine was hastily manufactured, and the vaccine died because people linked all sorts of maladies that occurred in the wake of the vaccine to the vaccine, which may or may not be attributable to the vaccine. And this is described at length in an article tweeted by Osama Bilal. But that's just, one slice of history. The next question is, once you ascertain the vaccine is effective, are we going to just give it to volunteers? No, that's not what we're going to do. We're going to have to strategize and think as we're ramping up supply. If, say, we can only have 1 billion doses in the next year, and we're going to need multiple billion doses, who are the people that we should give it? How do we distribute the vaccine? How do we stop spreading infection? Do we give it to people exposed? Which communities, which populations do we target first? These are serious public health questions. They're not the kind of question that is amenable to merely allowing the most vocal and ardent person who wants the vaccine to get the vaccine. That's not the right answer. I think that the potential for such a program to be abused by rich and well-connected people who ironically are in the communities that are the least decimated by COVID-19 is enormous. And this proposal will inherently exacerbate disparities if the vaccine were to be effective, but it might not even be effective. These criticisms were made by many, many people after reading these ridiculous tweets and blogs. Then one of the prime perpetrators of the amplification revised the proposal to say that what he actually meant was that we randomize the off-protocol use of the vaccine. If you're randomizing the off-protocol use of the vaccine, that's a non-idea. That's just running a larger randomized control trial. So I guess it's a stupid idea. It's that, they say there's no such thing as stupid ideas, but there are some ideas that are stupid. This was a stupid idea. But 5 years ago, 10 years ago, a stupid idea like this would not be getting thousands and thousands of retweets and thousands of likes and circulated so widely. A stupid idea like this would die quietly in a paper that no one's reading, or some sort of scratching that no one's reading. And I guess what I want to say is that, social media is super good at critically appraising new articles. It's super good at amplifying and clarifying and disseminating your work to a broad audience. But, novel ideas like this are really best suited for a proper formulation, not an 8-tweet thread about how it might occur, but a really fleshed out proposal for what you're saying, and even submitting it to a journal so that at least you get 3 other people to say this isn't totally wackadoodle. That it really is the better format for this. Somebody made an excellent point, which is that, even though this discussion is off base, there are some legitimate questions about whether or not we could have an adaptive randomized vaccine trial. And I thought, yeah, absolutely. You could have continuously enrollment. You could have multiple candidate vaccines tested in a single trial, just like UK Stampede, which we've talked about on this podcast. You could even bend the randomization ratio based on interim results, which there's so many Bayesians out there who'd be happy to talk you through. All these sorts of strategies are good. But again, these are best fleshed out in a full-length article, and it will be well published. It's a timely and clever article. They're not meant for someone who has only a passing familiarity with drug approval to be tossing out on the internet. So I guess part of my concern here is that this is a bad idea that got, massive amplification. The other part of my concern is that, what is going on social media? Why are so many people compelled to talk about things that are far beyond what they know or what they've thought about? I mean, in the beginning of the pandemic, there were many people who model pandemics who were rightly frustrated that the media was not including them in the discussion. They were included in the discussion. Now, here they are talking about hydroxychloroquine and dexamethasone. But since when does their pandemic modeling experience translate into judging the efficacy of therapies tested in randomized controlled trials? I think it doesn't. So, they've shown a bit of hypocrisy where initially they felt slighted because their expertise was not included in a broader discussion. Now we're past the moment where people are interested in their expertise, but they desire to cling to be part of the discussion, and thus they want to expand their expertise, which is ironically the thing they faulted others in the beginning. So it's a grand hypocrisy. There are entire Twitter accounts of people who know nothing about anything, who just say this is a COVID account. They may have a PhD in an irrelevant subject. They may have, some passing scientific credentials. They may have once used the restroom at Harvard just to say that, spent time at Harvard on their Twitter profile. I mean, this is really ridiculous. And I think the audience has very little ability to discern who actually knows anything versus who knows absolutely nothing on this topic. And I don't even know what their motivation is. I honestly am puzzled because they're not selling any products. They're not, they don't have any real line of work they're drawing attention to. They don't have a long publication record that they're drawing attention to. They don't have, in many cases, they don't even have academic careers. I don't understand what's the motivation. They get a couple of interviews on these god-awful cable TV shows that, to be honest, very few people watch those cable TV shows. They're not what they used to be. Television is not the same media it used to be. And to go on TV, no one will remember you were ever on it in 1 year. They'll never remember. No one will remember you were quoted in a New York Times story, a Time magazine, in 6 months. They'll never remember. No one will remember you went on TV in 1 year. Nobody will remember you. They won't think that you helped the pandemic. They won't think that you're a good communicator. They won't think about you ever again. That's not to say that there aren't some people who do a good service of communicating things, I think, clearly and effectively. Some people do. But I'm just saying that if you are not someone who understands what you're talking about, and yet you jockey so hard to be this person to communicate to the public without understanding things substantively, I don't know what's in it for you, honestly. You're not selling any products. You're not going to get any money. You're certainly not going to get any durable reputation or fame. You're going to get this illusory bump in your follower count and on some television shows that few people are watching. I mean, I just think that that's really, really silly. And the best thing you should do is just shut up. You should just stay quiet. We should think about what we're really savvy at talking about. I mean, my interest has always been, for at least a decade, the rigorous appraisal of medical products that are ingested by people. That's what I spend my time thinking about. And when we were talking about modeling, I did my best to maximally keep quiet, although I also read a great deal about them in the process. And I think that I and many others who were really on the therapeutic side, we were reticent to comment about models that we were not routinely making, even if the construct of some of those models in retrospect turned out to be grievously in error. I call this Twitter COVID opportunism. Scientists with only a passing affiliation with ID, pandemic response, drug approval, drug trials, vaccine development, vaccine trials, feel compelled to build a Twitter brand talking about COVID. Now, somebody pointed out that, COVID hits so many dimensions of our lives, we shouldn't really be exclusionary in who can talk about it. And I'm saying, absolutely not. I wrote a commentary called It's a Supernova in Medical History. We need historians and sociologists and all these people brought to the table. But what we don't need them is we don't need the historian to be telling me how drugs should be approved. We don't need the sociologist to be talking about the modeling. We need them to talk about what the expertise they have. But that's not what's happening on social media. What's happening is people are starting their little Twitter brand by talking about the little sliver they know a little bit of something about. And then the moment that the zeitgeist moves away from their sliver, they do their very best to read CNN the night before so they can regurgitate news stories and talk about things they don't know anything about. And the problem with talking about something you don't know anything about is you just amplify a canonical message. You don't actually inject any original thinking or original dialogue. In fact, that's one of the biggest problems with oncology podcasts that I have discovered several new ones and I've been listening to. And some of them are literally reading the abstracts of the articles in the major journals. That's not a service. I'm sorry. If you want to run an oncology podcast and you're just going to say, oh, new article out in JCO. The JCO article says that adverse events occur frequently in people with who are exposed to pembrolizumab. But if you retreat them, it's actually, many of them can stay on it. Okay. That's not an analysis. I mean, I haven't looked through that paper yet, but when I listened to this terrible summary of it, my first thought is that, very likely there are patients whom doctors are unwilling to re-challenge and patients in whom doctors are willing to re-challenge with checkpoint inhibitors. The ones we're unwilling to challenge are the ones in whom we think the adverse event was really attributable to the drug and really bad. And the ones we are willing to re-challenge, probably the ones we think, well, it might have been related, might not have been, and it probably wasn't that bad. And when you add that bias in a confounding by indication retrospective data set, you should not be surprised that of the people in whom you re-challenged, it was tolerated well in 2 out of 3 or something like that, or whatever it was in this silly summary that I listened to. But the real question is whether or not, if you mandated or took away from that paper the lesson that you ought to more often re-challenge, which is the causal question we care about, if in fact you will do good, or if in fact you will be knee-deep in AEs because you're going to re-challenge people who were not those re-challenged in the retrospective study. But the discussion of this article does not include people who will even think about the article to that for one step, just one step of thinking. And if you're not going to do one step of thinking, you're not going to do one step. You don't even want to do the one step. If you're not going to do that, just keep quiet. You don't have to talk. I just want listeners to know that I've done like 7 takes to try to put a more polite word in what I think people should do in this situation. So that's a aside. So if you're not going to do that, if you're not going to actually invest your mental energy in reading it and curating it and giving us your take on it, then you shouldn't just regurgitate it. And if you want to just be this regurgitation artist and create a show called Oncology Regurgitation, we regurgitate abstracts, or COVID Regurgitation, we just regurgitate news stories on COVID, you're not a COVID expert. You're not doing a public service. You're creating a brand that I meant to talk about this a little bit. This brand is the most foolish brand to create. Someday COVID is going to be in the rearview mirror. Even if it's an endemic virus, it's going to be in the rearview mirror because people aren't going to talk about it all the time, and interest in it is going to dissipate. And so 2 years from now, you build your Twitter brand on being the person talking about COVID, and you know what? People are going to be talking about the next thing, and that next thing might be blood-based cancer screening. It might be, I don't know, some tracker that you put in your nose and tells you everything about your thoughts. It's going to be something different. And these people, I assume, are going to try to pivot yet again and take their already stretched skill set and apply it to some space where they really don't know anything about and try to continue their brand. But I think it's going to be even more and more obvious that this is a disingenuous brand and this person adds no insight and they're just regurgitating CNN stories to you. So why the hell are you following them and not just reading the CNN article yourself? I think that's going to be evident. So I think this brand has no legs. It has no purpose to the brand. You're creating a brand that's headed like a lemming for a cliff. It's going to be falling off that cliff the moment people lose interest in this topic, which they will. It's fine to create your brand. I mean, I've been an advocate for that on social media, but the brand has to be like the thing that you're going to be studying and talking about and offering novel insights for like your duration of your career. At least if you want to purport to use your academic credentials as the reason why people ought to listen to you, it ought to be because you're offering novel insights. So anyway, I've been listening to some more oncology podcasts, and every time they summarize a paper and the person reveals, even when you summarize a paper in the abstract, if you have spent any time reading articles or praising articles, you'll know that there are all these pitfalls you could easily point out. They don't even mention that. I don't think they understand that. It's not a service. It doesn't help anybody. We'll see what happens. The listeners will decide. But, plenary session, that's what we try to do. We try to give you the opinion of somebody. We have a new disclaimer now. The opinions expressed on the show are those of whoever said it. That's the new motto. And it's really true because it's not the opinion of CNN packaged through the filter of somebody who doesn't know anything about vaccine approvals. So this vaccine approval fiasco, a manufactured idea on social media, gained a lot of traction by being tweeted by all these people out there who are ill-informed but desperate for something. Really a disservice, really sort of an embarrassment to the people who did it, and it's just not necessary. I mean, it wasn't in response to anything. It was literally coming up with something. It could have been fleshed out as a white paper, in which case, people would have seen how ridiculous it was. So, on that positive note, we're going to turn to the next segment. The Adora Study. I had to go back to Adora because there's been so much discussion and quote-unquote debates about Adora. Like all great debates, in many cases, the sponsor of the debate receives money from AstraZeneca. The moderator receives money from AstraZeneca, and all the debaters receive money from AstraZeneca. Thus, the debate is fair and balanced when every single party is paid by AstraZeneca. I think these debates actually kind of they are really kind of clever, because if they're real fundamental concerns and you stage a debate that's so far to the we should do this side of the issue, you create this sort of false sense that the two sides are, well, we should wait for the overall survival, or we should do this right away, or we should wait for the overall survival and do this right away, or do this right away without waiting. Extremely skewed adoptive positions, and that's what's espoused in these debates. These are debates that don't hit this issue as hard as it ought to be hit. And of course, when everyone is conflicted with the sponsor, it's sort of a pseudo debate. It's like a primary debate. It's the primaries. It's the primaries of one political party, and there's not yet been a national election to debate the issues more broadly. So I followed some of these debates, and I thought that they were laughable. When both parties are paid by AstraZeneca, they don't even acknowledge that patient groups are involved, patients are quoted, and they are also receiving funding from AstraZeneca. I mean, it's kind of a despicable situation, and yet we have to tolerate it, or people don't know any better. So, this is the Adora Study that I want to just briefly point out the substantive flaws of. A randomized trial in 1B through 3A non-small cell lung cancer, which is activating EGFR mutations, randomized to, after surgical therapy, the receipt of osimertinib versus placebo. And you could have chemotherapy, but you didn't have to, and the rates of chemotherapy appear to be extremely poor and beneath the standard of care. There are some problems with this study. We don't know them all because the paper is not yet published, but we have a sense. One sense is that we shall see, but let's talk about staging. If you're running an adjuvant study, you sure better have staged people appropriately. And for people with 2 and 3, it would be criminal not to do MRI brain and PET CT. If you only did a CT brain for a 2 or 3 patient, stage 2 or 3 patient, you're performing, that's criminal. That's beneath the US standard of care. And why does that matter? If you're doing an adjuvant trial and you're the sponsoring company, you want the staging to be lousy. The worse the staging, the more occult metastatic disease you're going to enroll in your study. And guess what? We know that osimertinib is probably better than placebo if you have occult metastatic disease. And if you don't do adequate staging to look for metastatic disease, you very likely are including some occult metastatic disease. Inadequate staging, you cannot post hoc figure out that the trial still would have been positive were it not for whatever fraction has occult disease. You don't know what fraction has occult disease because you didn't stage them. And you don't know what % of the contribution of the outcome is driven by treating occult metastatic disease versus just not treating it. So inadequate staging is probably, we will see, but I have a strong feeling that's going to be a major problem. Another problem, if a patient had 3A lung cancer, those often go to interdisciplinary tumor boards, and post-surgery they receive some consolidative radiotherapy. And there's some data that suggests that might have a benefit, and we can talk about the limits of that data. But it might be the case, I hear, that this trial has a prohibition on that. And if it has a prohibition on that, that's a big problem, because that's a prohibition on something that people think is the best thing you can do for patients, thus further increasing relapse rates needlessly in the control arm. The chemotherapy rate. We don't want a trial of osimertinib versus placebo in people who are being tricked to not get chemotherapy. And one way they might be deceived into that is to say, well, we have a trial that's a novel targeted drug, or placebo. It's targeted just for your cancer, for your mutation. But, you could still get it after chemotherapy, but, it's open right now and there's spots available right now and you don't want to miss it. That sort of attitude that could easily dissuade someone from chemotherapy. You want to max out chemotherapy, adjuvant chemotherapy, which is an intervention that has a durable DFS benefit before you start adding on TKIs, which may only delay the ultimate presentation of microscopic disease. The other question that will come out is this a curative treatment, or does this just delay inevitable recurrence? We already have this huge problem of when you give Ossi in the front line, you find yourself running low on options when those patients progress. Often you're going to chemotherapy alone. And when you're giving the adjuvant setting, what are you going to be giving them in the metastatic setting? And don't say you're going to be giving them PD-1 antibodies, because there's a big question mark next to that. You're going to be running out of options. And the real question is whether or not the routine upfront use to many people, some of whom are already cured, so they're just getting toxicity with no benefit, is better than giving osimertinib upon metastatic relapse in the fraction of people who relapse. Next, what drugs are given upon metastatic presentation? I have a sneaking suspicion that this trial is going to be giving people who present in the control arm with metastatic disease gefitinib or erlotinib. If they're doing that, this trial is not relevant for the people who are praising this trial. They have abandoned that strategy. The same doctor who's saying this is a practice-changing trial could easily be in a malpractice courtroom testifying, and they would say something like this: well, Your Honor, this oncologist didn't provide appropriate care. This was a stage 2 patient. The oncologist didn't get a brain MRI. The oncologist relied on a CT that was done for a different purpose a few weeks before. The oncologist didn't give the patient a brain MR, didn't give the patient a PET CT. The oncologist didn't place a referral for radiotherapy. There was no discussion of whether or not radiotherapy was indicated in 3A. For some reason, there was no adjuvant chemotherapy given, Your Honor. And then when the patient presented with metastatic disease, the doctor gave gefitinib, even though, in my practice and the practice of my peers, we switched to osimertinib for the last year. So, in other words, if the control arm of the study is going to be considered malpractice, and the PI would go to court and probably testify in a sort of a hypothetical case that is beneath the available standard of care, how can they then justify this control arm as proof that this trial is successful? This trial will have an OS benefit. If you don't stage people appropriately and you don't give appropriate therapy upon progression, there's no doubt there'll be an OS benefit, but it will prove nothing. It'll prove nothing. Many of these debates say the OS benefit is the tiebreaker. It's only a tiebreaker if you run a correct study. If you run a lousy study that doesn't assess the question that actually matters, OS benefit later is not a tiebreaker. If your control arm is essentially malpractice, then the trial is not a useful study. The real question is, why are all the big names in the field praising this study? And they praise the hazard ratio. The hazard ratio point estimate is 0.17 or something like that. They're like, I haven't seen a hazard ratio so good. It's a hazard ratio that's exquisite for an endpoint that is not a measure of people living longer or living better. Some people say, well, DFS is quality of life. No, quality of life is quality of life. And you can measure quality of life. You can power the trial for health-related quality of life. And if the metastatic disease that you're averting is really symptomatic, devastating metastatic disease, you will see that in an improvement in health-related quality of life. You didn't do that. Don't assume your surrogate measures quality of life when you can directly measure quality of life. That's just a classic, poor argument by people who don't understand basics of trials and trial design. And then the last thing is, one of the arguments I heard was, well, patients aren't looking for OS benefits. They're looking for things that keep us going to get, just in case there's some new breakthrough therapy down the road. And that's why, PFS and DFS are okay. I'm sorry, that is an incorrect argument. Just think about it. What is the prerequisite to receive some future wonderful therapy? Okay, let's just say sometime in the future, the doctors are going to come up with a magical curative therapy for this condition, hypothetically. Do I think it's going to happen for this cancer? I have my doubts, to be honest with you. But let's say that it is coming. It's coming for sure. What are the prerequisites to receiving this magical curative therapy? What are the prerequisites? Well, one, you have to be alive. So if you died before the therapy, you won't be able to get it. So that's true. Two, you have to have, a reasonable functional status, a good performance status. But to be honest, if it's really so wonderful, they're going to bend the rules. They're going to give it to you with PS of 4 if it's really so wonderful. And if you think that won't happen, then I don't think you've followed what's been going on with the PD-1 and PD-L1 antibodies. Okay, they'll give it to you if even with a diminished functional status, if they believe it's really, really good. And those are drugs that are not Lazarus drugs. But if it was truly a Lazarus drug, yeah, they'd give it to you. So you have to be alive and have some baseline performance status. Those are the prerequisites. Does it matter how long your first progression was if the drug doesn't improve quality of life? No, it doesn't. It doesn't matter at all. Drugs that improve PFS without improving OS do not keep patients going longer to receive a magical therapy that does not yet exist. That is a fallacy. Drugs that improve OS keep people going longer to receive a magical therapy that does not exist. Not drugs that improve PFS, because if you improve PFS and don't improve OS, your window on the back end to get this magical cure is not any longer. And yet, this argument is repeated by so many people who presumably have an ounce of intelligence, but yet they have not even thought about this. For 2 seconds, it would take 2 seconds to think about this to realize that it is a fallacious argument. Who created the argument? I strongly suspect it was somebody in the industry marketing arm created the argument. And then the KOL, while buying some doctor lunch one day, whispered it in the doctor's ear. And the doctor repeated it over and over because this doctor has no training in statistics or thinking clearly or epidemiology, etc., etc. So the real problem with ADAURA and these fake debates is that both sides are skewed heavily in one direction towards adoption of the therapy. There is no one participating in the debate not paid by AstraZeneca, and there's no one participating in the debate who's actually going to really rip this trial to bits. And that is a fake debate. And the best thing to do when there is controversy about your study is to stage a fake debate, because then both sides of the debate are terms you already agree with. It is literally a primary in a political election. You'll be happy with both sides because they're far to the right or far to the left of the other party. And that's what's going on here. And so, ADORA trial. When it comes out, you're going to have to look, what was the staging? If they allowed CT and not brain MRI, you should just rip that paper in half. If they did not mandate PET CT in 2 and 3, rip it in half. If when patients on the control arm presented with metastatic disease, they did not give osimertinib, rip it in half. If they ban radiotherapy in 3A, if they do not allow or potentially dissuade participants from chemotherapy, rip it in half. And then, somebody said, the hazard ratio is still 0.5 in 1B. Stop talking about hazard ratio, okay? The hazard ratio is the instantaneous probability of the event in one arm divided by the instantaneous probability of the event in the other arm. It is not a measure of the probability that you're going to do better or live longer at any point in time, and is not the measure of how much better you'll live longer or live better, and whether or not the costs and the toxicity justify that magnitude of benefit. If you do not know what hazard ratio is, you should not say, this is the most impressive hazard ratio. I do not think they know what hazard ratio is. I don't think they know how to use it. I don't think they know how to talk about it. And proof is that they're just so happy to get a 0.17. We've seen hazard ratios exquisitely low, typically with PSA in some of these prostate cancer trials. It often is lower than radiographic progression-free survival because you can generate a far more drastic difference in PSA that translates into a far less drastic benefit in radiographic PFS, which translates into an even less benefit in OS, which is actually, surprise, surprise, the thing that actually matters to patients. If you took somebody with prostate cancer and did an exchange blood transfusion, you get a hazard ratio of 0.0005 potentially for PSA. Just take all the blood out of him and put a full exchange transfusion on this patient. You're going to lower that hazard ratio, but you're not going to make them live longer, live better, because a hazard ratio for an endpoint that doesn't actually matter is not a useful thing. So ADORA, the real lesson of ADORA is this is a scam. The scam is that multinational companies can perpetuate absolutely god-awful studies that don't inform our actual patients, and there is not a single person who has taken the oath of a doctor who's willing to stand up and say that this is unacceptable, because they're all getting paid by AstraZeneca, participating in sham debates, and perpetuating the lower and lower standards. And then they go on Twitter and they praise each other for congratulations on your lousy study that doesn't inform our practice, and your lousy study, then your lousy study, and we all have drug approvals for lousy studies, and we have $150 billion expenditure on cancer drugs. We're using them early, often, and continuous. The drug companies are getting rich. We're getting a tiny pittance of the drug companies' wealth in terms of personal payments, and our patients are, unfortunately, not being cured of lung cancer. They're not living longer substantively with lung cancer. The years of life lost are still gaping for somebody with lung cancer. They're not living longer for the magical cure. That's just some erroneous argument that was constructed by a marketing person that the KOL who's taking fistfuls of AstraZeneca money and putting in their pocket doesn't have the brain power to tease apart from a clearly fallacious statement and has no ability to decide whether or not that's true or false. This is the entire problematic ecosystem of oncology. This is the book Malignant. It's written to draw attention to this fact that the core structural failure is all of this. We have lousy trials and drugs that don't do what we think they do. We have false and inflated beliefs. We have hype rhetoric, and we have a financial system that would be considered outright corruption and fraud in any civilized nation in the world, and it is all tolerated. And people pat themselves on the back and stage fake debates, and don't have the courage to have a real debate run by somebody who actually is not paid by AstraZeneca as a moderator, and somebody who's a panelist who's willing to rip this trial to bits. And we don't even have the trial yet. And yet, of course, it's already being hailed as a standard of care. If you did the right study, you staged everyone appropriately, you gave chemotherapy as much as you could give. If the interdisciplinary tumor board felt that radiation was indicated in 3A, you would give it. And you randomized patients with lung cancer to osimertinib or placebo. And for the patients getting placebo, you followed them closely, and at first sign of recurrent disease, we gave them osimertinib. I would be very confident that osimertinib does not improve overall survival and does not improve quality of life. I bet you're adding tons of toxicity, and you're adding tons of market share for this drug, and you're deteriorating quality of life of 100 people. The fraction that's already cured, they're just suffering a quality of life decrement. The fraction that has a delayed recurrence, they're not getting a quality of life benefit, because often with really good follow-up, you can find recurrence very small, and osimertinib is well tolerated when you get in the metastatic setting. So I think you're just getting the negative impact of more osimertinib early, and I bet there's a huge quality of life decrement in second and third line therapy if you get this in the adjuvant space, because you've exhausted EGFR inhibition, and you're going to be giving the patient chemotherapy, and they're not going to be so happy about it when they do have declared disease. And so I bet if you did the right study, you would have no OS benefit and probably a net quality of life decrement if you measured it across the lifespan. That's the irony of this study, is that it's so misconstrued and so many people are getting their palms greased by this company that it's going to lead to the erroneous conclusion. I bet it would be the other way around. I don't know that for sure, of course, because they're never going to do the right study. Because why would we need to do the right study when there is no pressure from anybody in this system, when everyone is on the take from AstraZeneca? Why would we ever do the right study? And then when we ask ourselves why cancer patients have such disgraceful treatment, and this is a disgrace, when you debut 71 consecutive drugs and the median improvement overall survival is 2 months, that is a disgrace for drug approval. When you're spending $150 billion global expenditure annually and the drugs that you're spending it on are marginal mediocre drugs that add 1 month in survival in idealized patient populations and maybe 0 months in survival in the actual setting, that is a disgrace. That is a society that takes a massive amount of human capital and then they say we're going to help cancer patients with it. And instead of actually helping cancer patients by providing nursing care and meals delivered to them and laundry service and things that would actually make somebody sick and dying with cancer have a better quality of life, we're going to pay for these horrible drugs that barely improve survival even under idealized circumstance. And we're going to give that to you. And actually the cost of making the drug will be pennies and the profit margin will be just diverted to other people. And that's really what we're going to do. That's the system. And that's what you see here. So ADORA study, I think that these are not limitations that are okay, acknowledge limitations and then let's just do it anyway. These are limitations that are damning. They're so damning. It's like catching someone doping in the Olympics. You got to strip them of the medal. And if you're not going to do it here, you're going to allow this and you're going to allow the next one to slide and the next one to slide, then don't be surprised if in 20 years in oncology, the majority of cancer diagnoses are still death sentences. Yet we are having global sales of $500 billion in cancer drugs. And people aren't really living substantively longer, but we feel like we're living longer because scans are getting so advanced. We can find you have cancer 5 years before we otherwise would find it. And so you get that nice lead time bias. I mean, I wouldn't be surprised if that's the case. And so I think bad study, poorly designed, and the failure are with the, we can talk about the failures, but I actually don't blame the company that much. These are their incentives, the incentives that they exist with, and company's a tiger, and when the tiger kills an animal and eats it, you can't blame the tiger. That's the tiger's motivation and tiger's goal. The failure, I think, are the regulators who have capitulated to the tiger. In fact, most of them are looking for jobs working for the tiger some days, so of course they're not going to be too harsh, and we proved that in a BMJ paper. And the other failure are the academic quote-unquote experts who surprisingly have no expertise in actually appraising trials, no expertise in actually thinking about how you would design the right study, no expertise in actually standing up for patients, no expertise in appropriate post-protocol therapy, no expertise in staging, conveniently no expertise when they're receiving payments. These are people who are doctors who, did take an oath, and that oath does include some things that aren't this is the new modified Hippocratic Oath. I promise to be an oncologist and I will always say that everything I do is in the best interest of patients. However, I understand that for-profit companies wish to get drug products approved, and whenever possible, I swear to randomize patients to control arm regimens far beneath the care I would normally provide, care that I would blame the fellow or even testify as malpractice in a court of law. I will permit in a randomized study if it allows the company to make money. This I swear in front of my teachers, the new modified Hippocratic Oath for an oncologist, as long as, of course, I'm receiving personal financial payments from the company. So on that positive note, we're going to turn to the next segment. Okay, I'm back in plenary session end of days bunker, joined via Google Meeting with Audrey Tran. Audrey is, of course, famous for questions from a medical student. It's great to have you. Great that you're willing to do this remotely so we can continue this great segment that everyone loves.
Well, thank you so much for having me. It's always so much fun to chat and just kind of think about these things.
Your segments are popular, but really? I think they get me into a little bit of trouble.
Really?
Yeah. I still get, like, every other day somebody messaging me and they're like, he doesn't think soft targets are as important as hard targets. I'm like, oh my God, come on, let it go. Of course I don't, but I discussed it in the episode, why I don't, in one of our episodes, episode 2.30. You can listen there if you want to feel outraged. But today we're on to something new, Audrey. So what are we talking about today?
Yeah, so today I was kind of going through, I feel like because we don't work in the office anymore,
Mm-hmm.
I was like just going through some of the tweets and other things we kind of discussed, and I found some really thought-provoking tweets that I that you could expand on, but it's something I kind of wanted more clarification on.
Mm-hmm.
And I think it was the topic of the fake pearls or the witnessing, I think, of these
Yeah.
pearls being formed in real time. And you have a tweet about this, I think, which people can look at. But ultimately, some questions you posed were like, what motivates someone to say like this is something you should be doing in clinical medicine? How do these ideas perpetuate? And how personally have you seen it developing either in clinical practice or just kind of on social media, so on and so forth? Because ultimately, I guess to me, the underlying question for that is um, the idea of credibility and authority, especially in times of uncertainty.
Mm-hmm.
And like who, kind of this larger question, like who's reliable and who do we turn to in these times of uncertainty?
That's a good question. So I guess should we start with the Pearl discussion and then go to
Yeah,
this
let's
answer?
maybe describe kind of what use like what that tweet was, and then kind of what motivated you to kind of think about that.
Yeah. Okay. So I guess whenever you do like clerkships and clinical clerkships, like we all get people giving us pearls, things that little tidbits of information they think are really really good. And I just mentioned that there's a tidbit I got many years ago, which was when you do a thoracentesis, you shouldn't take off more than 1,500 milliliters. Um, and the reason why is if you take off more, the patient will be at risk of re-expansion pulmonary edema. And I had heard that, and then there's so many different pearls I've encountered over the years. Um, Another pearl somebody told me was, I washed my hands and I entered a room, and I set up the kit for the central line, and I got everything ready. And then I was about to put the sterile gloves on before doing sort of the final steps before I started the procedure. And then somebody said, "Hey, stop right there. You got to wash your hands." And I said, "But I just washed my hands when I entered the room." And they're like, "Well, you have to wash your hands yet again before you put on the sterile gloves." And I said, "What's the reason for that?" And they're like, "Oh, it's part of the checklist guidelines." I was like, okay. So that's another pearl. And then, sometimes you're curious and you start to think to yourself, this is an impermeable sterile glove. And I never touch the outside. I only touch the inside of the glove as I pull it on my hands, because the cuff is flipped out.
Mm-hmm.
And so why is it again I have to wash my hands when I've literally washed them like within, 2 minutes ago, and I've only literally touched like the sterile plastic of the thing itself? And then you start going down a rabbit hole. The same for re-expansion pulmonary edema. I went down a rabbit hole. And the rabbit hole is, where did this come from? Who thought about it? And I think so often in medicine, the deeper you go down that rabbit hole, the more dissatisfied you are. And the more you end up thinking that somebody's pearl is a cheap plastic kind. It's just a cheap plastic pearl that, doesn't really carry any weight. And I just feel like that's true often. And I guess the comments to that tweet generated so many people had like cheap plastic pearls they were given, and they kind of talked through what those pearls were. I guess I've always been interested in these pearls because I'm interested in the psychology of, as somebody who's teaches, not always formally, but sometimes informally. I guess I'm very hesitant to say things that I myself have not, really dug into and verified. I think that so much of my teaching, like when I'm on service, is, I ask students like, or I ask fellows or residents like, what randomized trial or what's the data supporting this? Something that I know really well, so I know it. I know it well. And so when I ask them about it and then I tell it to them, I'm confident that I'm not misleading them. But these other kind of pearls of like, how do you do something, or, sort of mnemonics or rules of thumb, I'm very uncomfortable passing forward unless I've verified the authenticity of, so to speak. And I think, my tweet was really about that, was about like this inauthentic pearls. And then the reason I thought it was so interesting in the time of COVID is, I really start to see these pearls formed in real life. I'm privy to some discussions where people were like, "Oh, what do we do with chemotherapy patients who are coming in? They might have COVID, but we don't know it. They're not symptomatic. Should we test them for COVID before we give them chemotherapy? And then, of course, the answer is, well, if you do a PCR test, you'll have to wait several days. And then, well, maybe we should have them come in the week before, do the PCR test, and then come get the treatment. And then somebody else might argue, but, then they could develop positivity between cycles. So how often are you going to treat them? And then somebody else might say, well, we want to know they're negative when we treat them with chemotherapy. Then somebody else might argue, but why do you want to know they're negative when you treat them with chemotherapy? Well, the virus would be worse if they have gotten chemotherapy. But then you go into, but is there actually any evidence that folks who are getting chemotherapy have worse incidence of the virus, or that it's a risk factor? Has it happened? What's the rates of it? Is it logical? And then finally, of course, there's, is there any proof that this is of benefit? And you can see that something that just starts sort of like an innocuous question, which is like, should we do this test before we do something? With best intentions, it just starts as a question. It so easily can get a life of its own, and people can come up with all sorts of logic and reasons why that is the case. And one can imagine, in a very short period of time, a university has set up as a policy that every patient getting chemo has to have a negative COVID PCR 3 days before. And of course, there's a harm to that, which is now you got to bring in people an extra visit, and you got to instrument them. And in fact, potentially there's a risk of nosocomial or iatrogenic sort of injury or spread of the virus. So, are you doing a net good? And anyway, the reason I thought of this was that's how these things are formed. I was like, how did it even people get this idea that this 1500 cc's, which I'm not sure I stated yet, but when I look into that falls apart really quick. And I gave a link to an article that really goes through some of the history of it and shows you why that's kind of neither here nor there. And I guess it's interesting to me, the psychology that goes into where these pearls get formed. And I think it's probably like an ancient thing. Like, for thousands of years, human beings, when faced with uncertainty, things they're fearful of, have formed pearls. And probably in the ancient times, a lot of our myths or mythology, a lot of the sort of heuristics and rules, they were formed under the exact same sort of social dynamics as the pearls of today, which is, do people need PCR negativity before you administer cytotoxic chemotherapy 3 days later? I can see that sort of human psychology. And for me, it was sort of unique because in my life, I came after the HIV/AIDS epidemic, and what I'm trying to say is I don't think there have been many diseases that have sort of originated on my watch. And COVID has, and that's given me sort of this unique insight. But I think your questions go even deeper. So tell me the next part of your question, which is about really credibility and who's credible. Yeah.
Yeah. And almost in response to just one of the pearls that I remember, one example before we go into that, what you're talking about reminded me of this thing you were describing in one of your cancer talks about the definition of a response rate, or the calipers, the carpet. With
great example, right? Yeah.
And to me, that was such an eye-opening moment, because I just kind of took the definition when it was in the NCI or some sort of definition based on some sort of official website. I was like, okay, I guess me being very oblivious to like this idea of like how definitions are set or standards are made in cancer medicine, I was like, well, I guess that means that must correlate to some sort of benefit for a patient. So I don't know. That's it. Like that percentage seems like 30% seems like a reasonable amount of shrinkage or whatever. So that must be why they chose that.
Right.
So
that's a great, yeah.
That's what they could detect with their crude instruments.
Yeah.
I don't know. It's kind of like when you go down that rabbit hole and you keep asking, why and why and why, and you come to this answer. It's not, I don't want to say it's anticlimactic, but there's some sort of, there is actually some sort of kind of humor in the sense that it was just the realization that we're all just we're doing our best and we're trying to figure it out, and given the technology at that time, or given that thing, and it certainly helped that Charles Martel was, a pioneer in his field, so that's why it was very much accepted as a standard. But I just always think of that example where it's like, I'm sure that many things in medicine could originate to one person starting it this way, and therefore they adopt it as a standard. And sure, there's improvements, but they'll always be credited for like that one thing. I don't know, just...
No, that's a great example. And let me just, to give listeners a little context, they can read about it, of course, in Chapter 2 of Malignant. But, in oncology, we talk about response rate so often. And, of course, we know the response rate by RECIST 1.1 is a 30% shrinkage in the diameter of the tumor. And then the question comes, why is it 30%? And I, like Audrey, I think when I first entered oncology, was like, well, that must be for good reason. And I just assumed it must be for good reason. And then finally I went and I traced the history as far back as I could. I ended up at a 1976 paper where I learned the origin of that size cutoff. And it is sobering to say the least. It's based on what, 16 men could feel marbles through foam rubber with calipers. Men in the 1970s, they're all men because, of course, that was the gender bias back then, could feel marbles through foam rubber. And from that information, from those humble beginnings, these cutoffs that were really operational cutoffs, they were cutoffs designed for an era of feeling marbles through foam rubber, which is supposed to be tumors through skin in an era before CAT scans, that has been codified and passed forward, polished into a plastic pearl. And so that's a great example. I hadn't even thought about that. Yeah. And then so when people tell you that, response, it's a great surrogate of benefit. We have a drug with a high response rate. And you really start to ask, well, does response predict living longer, living better? And you see those studies are kind of weak. And then you wonder why. And then this is the kind of stuff you find. Yeah, and I really think that the other podcast I think about is Bedside Rounds, where, Adam Rodman goes into the history real deep. And every time I listen to one of those, I'm like, oh boy, that's a fascinating tale. Yeah, but yeah, go on. What you were going to say about credibility? Yeah,
and so I guess just it's one of those things where I find that in this world where there's just so much uncertainty. It's there must be some sort of need for someone to be right, to have, and not just that it's that there are facts. I feel like how you can only have facts if they're verified or proven by someone or supported by someone you trust.
Yeah.
So I guess I don't know, just this larger conversation of what does it mean to have credibility and authority? Who has it now? And how do we think about this in a way that's constructive and not just nihilistic, and just being no one trusts nobody, but at the same time it's is it the most charismatic person in the room that seems to have the most credibility, or is it someone who's really articulate, but they could be just spewing, nonsense?
Right.
What about
Such
people who are have prestigious titles,
Yeah.
and so that lends people some sort of credibility, and I just feel people are so willing to give their, I don't know, they just want to be able to trust and believe somebody because it's easier than having to do the work of verifying
That's
everything.
the issue. Yeah. I think it's because so that's so good. So I guess I would say maybe to take this in two parts. One part maybe we can think through is what are the things that you see that give someone the air of credibility, whether it's true or not what they're saying. And it's exactly what I think you're saying, which is one, prestigious title, venerable institution. A new study from Harvard Medical School finds, or Dr. So-and-so, the such and such professor of medicine at the Stanford Medical School. That sort of title lends credibility. I think charisma lends a great deal of credibility. I mean, people who put things well, who are jovial, and it's less so in maybe impersonal situations like Twitter and on television. But you're in a dinner party with somebody, and it's often charisma that carries the day when you discuss sort of when you're having dinner with a bunch of people and you're discussing a social political issue or something. Charisma in those sort of intimate situations, I think goes a long way. I think one of the other things that goes into what we perceive as credibility is the how many times you hear something, how many people are saying the same thing. And that to me is what I see with oncology and I think the industry comes up with some talking points, and then they have all these KOL meetings, and then they kind of, I don't know, use money to grease the wheels of getting that idea in everyone's head. And then I go to I go to these meetings and I see so many smart people repeating the same thing. Like, oh, because there was crossover in that trial, that's the reason why there wasn't a survival benefit. And I was like, yeah, but that's also potentially a reason why a safety signal was masked as well. I was like, you're not independently thinking through that idea. That's an idea that somebody passed to you. Right. It's a plastic pearl you're passing along. And I think the industry has been able to turn up the volume, especially in oncology. And then I think the next part of your question is like, do you have to verify everything yourself? And then the last part of credibility. So the how many people say the thing. I also don't like scientific petitions. One of the things that I think is kind of trite is there's a petition going around that says 100 respected scientists believe that universal cloth masks is a good idea. They get a petition. Or 700 scientists think we should abandon the p-value cutoff of 0.05. And the reason these things trouble me is that, come on, these kind of decisions are not, it's not a vote. Science isn't just this vote of what we all think is plausible. I mean, if you really want to persuade people that the evidence is good, nothing stops you from doing like a really good randomized trial. If you really want to persuade people that we should abandon statistical significance, you could do a cluster randomized trial of journals or articles and actually abandon it or not and follow those articles in time to see how many people have reached different conclusions and how they lead to different behavior and kind of show that, one way or the other leads to a better interpretation of studies. You could do pilot studies. I mean, what I want to say is you can't confuse people believing something's true for evidence that something's true. And I don't like those kind of, I try not to sign these petitions, although people email me from time to time. So I guess like your answer, like and then like what is real credibility? I think that more and more as I get older and do this, I try to verify things myself. I try to hear what people are saying, think about it fairly, try to be in a place that's dispassionate. And I just give you one example of like, I don't know, a claim that's been made. There's that seroprevalence preprint from Santa Clara that's getting like a lot of news coverage. And it seems like there are some significant problems and issues with that. And I think those should be investigated. I mean, I'm not gonna, want to brush that under the rug. I think that should be investigated. But one of the claims that was tossed out on Twitter that's, I thought, very provocative was that the authors sort of intentionally produced a lousy study so that they could accomplish a policy goal for a political purpose. And the policy goal is to lift lockdowns. And so, I mean, I had heard this kind of claim being floated in a number of streams that directed at me. And so then I was just like, okay, that's kind of a bold claim, not just the usual somebody did a lousy study, which is the, Monday, Tuesday, Wednesday, Thursday, Friday kind of thing, but that they had sort of a sort of, bad agenda, and then they started to do this. So then I thought to myself, how would you evaluate such a claim that somebody's like did a study so bad because they wanted to shift the tide of sort of policy and intentionally? And I guess, well, I don't know how to do a study at all, but one way would be, what if you took every study that ever looked at this question and plotted it out and looked to see where their value fell? And if their value really fell as an extreme one end or the other, then I think you've got sort of prima facie a case that they're trying to influence what people think because they're pulling the field in one way or the other. And so I did that. And what I found was that this study is, and, people can debate, but somebody argued with me, I said it was in the middle, but they said it's not in the middle. I was like, look, I mean, we have to agree that like when you look at something, this is like roughly in the middle. I would say it's like at the 40th percentile of studies. And I guess to me, what that means is, I guess I put a question mark that a study that gives a result at the 40th percentile could shift the direction of a ship, because it is in the middle. If you want to shift the direction the ship's pointing, I think you have to have the most highest number or the lowest number, right, to really pull something. If you shoot something in the middle, how are you steering the ship? I mean, it doesn't make sense to me. And the reason I did that exercise was, like, I mean, I think it speaks to your credibility, which is like, I wanted to evaluate in my own mind the claim. Is it plausible that these people had an ulterior motive? And I can't get in their mind, but one, I think, intermediate is, if you had an ulterior motive, I would think you would produce a very skewed estimate. So ergo, is this estimate skewed? So like, that's kind of like what I wanted to say was, I wanted to like do some work on my end to evaluate something that people have been telling me. And the same is true for, anything from like, should you pull off more fluid than 1500 cc's? It takes work. You got to dig through all these boring references to find. But where were you going? You had something else, I think, another part of the question that I'm ignoring.
Yeah, so I think that's it's a really interesting point where it's like, I think, it does take, I don't think people realize how much effort or how much skill goes into practicing verification or just practicing like researching on your own.
Right.
I always think that's a really interesting point where it's like, at least I may never, I think there's an acceptance that you may never get to the full truth, right?
Right.
Because it's like the tension, right, with what is really the kind of the point of debate. But then that's why, and in those moments where it's like, when you can't reach that absolute truth, like an intermediate or some sort of surrogate will have to do.
Right.
But then also just like transitioning off to this point where it's like talking about controversy and talking about this idea of, especially when in times of uncertainty, nothing. Like, I think everyone is still vying for I think not control, maybe control of what is truth, or what should happen, or what should we be doing moving forward. And I guess I wanted to kind of bring up this kind of counterpoint of when people weaponize credibility. How do we stop? Like, I think it is really important to question credibility and always check your sources and do the verification. But I also think that it must be done in a way that is with the intention of again arriving at the truth, and not using it as a way to silence people, especially women, people of color, people who I don't know, maybe just are in a minority of opinion, but an opinion that is truly debatable. Truly is like, we don't know how to move forward, and even though it seems more popular to do this, like I don't know, I think that, is it enough to say that a fact is not credible though? Like when we're talking about attacking credibility. Yeah,
no, I agree. Like, how do you do it, I think, appropriately, attacking credibility or attacking a view? And I think, it also reminds me of the recent thing where, I saw that, these Santa Clara authors, they put out this paper, and obviously it comes out with this idea that COVID's the number of people who have COVID, who die of COVID, is a lower fraction than what people think. But when you plot it all out, I actually think it's more or less where a bunch of studies are falling. But people went after them hard. And, I do think that they're, like, at times comments do go a little bit below the belt. And one was, one of the authors in a video was wearing a white suit, and somebody was like, "Look at him wearing his stupid white suit." And I was like, "Come on, man! You're busting the guy for wearing the white suit." I was like, you really don't realize that you're like crossing a line, like when you're like, "Oh, and he's got his goddamn white suit on. Look at this smug guy in his white suit." And I was like, "Oh my God, come on! Let's just keep it on this clean study." But your point about the types of people who are silenced, I think, is a point that I didn't do a good job of making in some of the things I've been saying about this, but it's a point that I really want to make, and maybe I will go back on Twitter and say this, which is, when you look at some fields like in medicine, neurosurgery and cardiology, or orthopedic surgery, the percent of women going into those fields is very, very, very poor. I think in cardiology, it's like 15, 16%. In neurosurgery, it's like, I don't know, close to that, less than that. In ortho, it's like 6%. And I think, I'm not sure that anything explains it. Like, it's not the hours, it's not the variability in hours, it's not, because there are women pursuing surgical fields that have a lot of hours, right? And I don't know, it's been fully studied, but one hypothesis, I think, is that some of these fields have a bro culture, a culture where it is mostly men, mostly engaging at a certain level of sort of discourse or dialogue, and it can be like very hostile to people who don't want to engage at that same level of, I don't know, combativeness or assholeness, if that's the appropriate term. And when in this whole Santa Clara thing, where Jeff Flyer and I wrote this op-ed where we say, like, gotta be careful to, like, argue with his points, but don't demonize the people, because we want, right now there's a lot of uncertainty. We want to hear different ideas. We want to know what people are thinking. We want people coming to the table. People are like, well, they don't appear to be silenced, and if they're willing to go into the arena and say things that have implications, they got to be ready to get pounded. And I was like, well, you might not actually get these three people to stop talking. They might keep talking. It's the people who watch that who are not going to talk in the first place. And that might be, I don't know that this is the case, but that might disproportionately be women. It might disproportionately be people from underrepresented minority groups. It might disproportionately be people who are younger, who feel vulnerable. They don't want to offer an opinion in a climate where anyone who offers an opinion outside of sort of a canonical norm is going to get stomped on. And it might be, and that might also be the same thing, like why ortho and neurosurgery have like such a low fraction of women going in. Not the hours, it's not the work. It might just be that you're creating a climate where it's not very welcoming to other people because of the way in which, you're treating each other. And I mean, people may not know what I'm talking about. I'm talking about, I had people tell me stories of like this resident didn't show up to work, so he like drove over to his house, knocked on his door, and grabbed him by his collar and said, "You think you're sick? Get yourself to work." That kind of like level of like, just totally crossing boundaries and like not respecting people and that kind of stuff. That's a hostile kind of culture to work in. So anyway, to your point. I always see this claim where it's like, somebody's like, "Oh, prove to me someone's been silenced." I was like, that's a very difficult. I don't know what you're looking for here. You prove to me somebody didn't speak up when they wanted to speak up. I was like, well, that would be a sort of a hard thing to show. I'm not sure it leaves a mark. But I think you're asking the right question, which is, like when you attack somebody's credibility. I don't like to attack like their credibility as a person. I like to attack like all the points they're saying, and I think you got to keep it focused on that. And then the last thought I'd for you is like, it's so funny that I was just telling somebody that like I feel so strange that I'm somebody who likes to call it as I see it, and I'm very blunt about that. And but even in like the last 5 years on Twitter, it's really, I think, devolved into just like total chaos, just total people looking to get angry about everything you say and just constantly, just ready, like they want to be angry and they want to attack. And there's so many more people than before. So I think it's getting to be like a very nasty place, as somebody would call it.
I had a lot of responses, lots of good stuff. Especially this idea of culture and psychological safety is kind of what the thing I keep thinking of being able. I wish I could do a study on how many times people have wanted to say something but haven't. I know you can't, I'm trying to think of but I do believe at the heart that is where so many missed conversations go, or opportunities for growth and conversation. But it's like if the culture is not permissive, in any workplace culture is not permissive to do so, those are the ones that are more resistant to change, I think. But I believe that change is some sort of loss of some sort of way of being. But yeah, so that's one. I think secondly, I think your point, I know you've made this a couple times, but I think it is so true, is I think we have to start thinking beyond just the two people engaged in the debate, but to realize that there's a community, and not even a stage, because I don't want it to be performative, but it is a stage. People are watching. It is kind of the stage where it is the hearts and minds of people who are undecided, or people. And I think it was someone had this tweet about there's a Nature paper about anti-vaccination sentiments, and just kind of like looking at Facebook groups or something about how even though pro-vaccination, there's higher numbers in the pro-vaccination group, the rate of anti-vaccination ideas are spreading faster. There's more heterogeneity, and so they're just kind of coming from all sides almost, and at some point, I think in 10 years, they might overtake pro-vaccination, just because even just the very clear message of vaccines save lives and vaccines are good, which is true, but also it doesn't seem to provoke the same sort of sentiment or community or whatnot. And so there's just a bunch of undecided people in the middle. And you kind of saw this graph of like, I don't know, these little, like, and I was
but
like,
on that,
wow.
I wonder if you share my view. I mean, I guess when I look at this vaccine stuff, I think that's a great example where it's like, it's getting so hostile. I mean, what do I mean by that? Like, look, I'm a doctor. I'm getting all my vaccines left, right, and center. I'm giving all these vaccines. Okay, come on. I'm in the vaccine camp. Like, vaccines are good, generally. Are there a vaccine here or there that we can debate the evidence and okay, yeah, of course. But, the childhood immunizations, good stuff. Okay. We're not, okay. That's where we start. You got some people out there, for whatever reason, this has become something that, they're gonna not like. And maybe it's just a way to express frustration about a bunch of different things. Maybe autism and the lack of social services for that is part of the frustration. I'm not sure. Maybe there's all sorts of other sort of social things that this has become a conduit for expressing frustration, which is, I don't want to do this. I don't want my kids to do this, that kind of thing. And maybe some of it is just being ill-informed, and some of it is like bad actors like Robert F. Kennedy or whatever, that Kennedy who's always saying things that are wrong. I'm going to watch my words. Maybe some of it's that too. Okay, but then like I just see people on Twitter like who make a career out of like being pro-vaccine, and by career make a pastime of just talking about it all the time, and it just gets like more and more hostile. Like, anyone who disagrees is just stupid and dumb and blah blah. And I think that's like, I'm like, how are you trying to win this debate? Like, and then like every once in a while somebody says something bad and it's just like attack, and there's like so many people attacking on both sides, and I'm like, this is just getting so polarized. It's like, I almost think it would be better if no doctor and nobody talked about it or tweeted about it, and they just like silently just did some legislation that says you can't go to kindergarten unless you get these shots, or you can't go to grade 6, and then just started to like just have some education campaigns and like let's just take this down a notch. I don't know how you feel about it, and like you often are in groups of people, like students who are very passionate about this issue. Do you think it's getting too heated, or I don't know, maybe it's just me?
I mean, I guess I'll be honest. I've not seen. I mean, I think I've mostly seen the hostility from anti-vaccination.
Oh yeah, they're hostile too. Yeah.
Yeah, but it's like I don't doubt that there could also be this. I think there's a sense of frustration.
Yes, on both sides. Yeah.
On both sides. Yeah. But also it's like there's sort of frustration that like, by communicating, it's almost like the fallacy is like, if we've communicated and we have this excellent campaign, why isn't it working? Or why haven't we convinced people? Or why does it feel like we're not winning the information war, Yes. And so like there's a frustration on that side when it's like I almost think, to me, this is like the fallacy of science communication is that I don't think you can just present the facts even in a clear, like that's the baseline. That's like the standard prerequisite for like communicating science, is that it's true and that it is easy to understand.
Yeah.
But but I think your point of view of like just understanding the psychology of people right now, socially on a larger scale, of like why are people who don't believe or hesitant about it? What is their concern? Like, to me, it's just like, this is, I'm extrapolating and I have really no way to test this hypothesis, but it's more just, I feel like this is like one aspect of agency, and in a world like, I think there's so many things that can be stripped away, especially for Americans who are very proud and like they love their freedoms and whatnot. But it's this idea, this is very obvious, clear in their face issue that feels like parents want to exert control over their kids. Like it's such a personal issue of like what your kids do and like how you raise them and what choices you make for them on their behalf.
Right.
And it's funny because it's like, it's not like I feel like maybe people also don't want to get vaccinations for themselves, but I think it's like this idea that
they're exerting control against authority on their own children. It's like a place they can, and to me, but it's interesting to me because America once was, a place with very, very high vaccination rates. So I don't think it's like speaks to that American independence streak. I wonder if it speaks to the fact that, all of the things going on in society, the gaps between income inequality gaps, wealth inequality gaps, increasingly politically polarized culture, and in the middle of all that, this is a place where agency can be exerted where you can really take control, and there's some sort of deep wounds that this is kind of covering. But I don't know that. I mean, I don't know the path to fixing it all. But I do see people going crazy on it, especially the anti-vaxxers, but also even a few people who are pro-vaccine just going crazy about it, and I'm not sure that's helping. I'm not sure it's a thing where, like, it's like I'm not sure one day you're going to get somebody, you can give a paper to somebody. If you're really in the anti-vax camp, is there any piece of paper I could give you and that you read and you're like, oh, I was wrong? I don't think that exists. I don't think that exists. That does not exist. It's not a rational decision. Emotion. I don't know. Or go on. What are you gonna say?
I think that's what I'm saying is that, to be honest, it's like, it's almost like, I think we have to be careful that we're not being elitist, right? It's like, I taught you and now you've changed. You know what I mean? Like that's the narrative of that sort of a thing. It's like, again, I don't agree with their decision about the outcome of people like not giving vaccines to their kids, but it's like, to them, this is like, they're fighting for their dignity. You know what I mean? And I don't want to, just because we can understand it doesn't mean I like enable or encourage that sort of thinking, if that makes sense. Like, I feel like there's a line that should be on. It's like, I think we have to like, it may be uncomfortable for us to like sit in that particular way of thinking if we don't personally believe that is healthy or safe. But I think that, again, this has to be an emotional appeal at some point, right? Because it's like, what landmark study like could I give you that would be like, suddenly I see the light, like, especially to someone who maybe doesn't have a background in like reading literature, or it's like, what's the difference between this PDF and this PDF? The colors, like the font size, I mean, it's at some point, it's not what you give, but it's like how much you are receiving input from that community, of any community that you don't agree with. I guess.
Maybe COVID will be the, no, that was well put, but maybe COVID will be the thing that can heal it, because when the COVID vaccine will come out, maybe it will, and I think there's a lot of people wondering what's going to be, how many people are going to want to take it. But maybe the way to frame it is to say, those times in life where you roll up your sleeve and you do something for your country. And I know a lot of you out there, you don't want to do it, but you got to just do it for everybody. And maybe, I don't know. I think it's going to be an interesting challenge, but but I don't know. Crushing one more rogue anti-vax tweet on Twitter, as many MD, PhD, fellowship trained scientists think is a worthwhile pursuit. I just don't think it's not moving the needle on this issue. All right. So this was a good discussion. Our time is winding down. I guess the overall themes were fake pearls, how are they made, credibility, the need to do more independent work, and I think this key issue of who is being silenced, and what is kind of the culture that's being created, I think is an important question. Yeah, these are always good questions. What are your? I'll give you the closing. You're the guest of honor. So what are your closing thoughts?
Sounds good. Yeah, I think I would say, I think those are all the main points that you talked about. But I think ultimately it's how do we question credibility and do a productive, meaningful conversation. I think always the end goal is like, do we feel like we've arrived at a point? And I think, like you said, this needle, it's funny because you're saying moving the needle, but I truly think it is like a three-dimensional sort of needle of like all these different parts. And I don't know, I think, like you said, I think we're all in stages of grief right now. Whether, and it manifests in very different ways for some people. And so, like you said, the chaos and the anger and the, sometimes honestly vitriol, or like just even the need to feel snarky, or like, no one's immune from like that feeling of like, we're all human. We're
all human. Right.
And I think the more we can kind of accept that, and embrace that, like, it's like you can get it out of your system, but I think ultimately understanding that we're navigating this uncertain time with a lot of different ideas, and how do you bring those ideas to the table you can't send, like, you need to encourage that, which is to say, like, the psychological safety component of, like, if, and also I think being brave enough to, like, tweet it out, which is always why, I just appreciate, like, you'll always, like, tweet something and I'm like, okay, like that, yeah, that's a good point. And then, like, I think it fosters discussions, or at least makes people think, and that's important. And then also just the idea of, I don't know, just when one idea becomes too dominant without, like, self-reflection, it's just like, how do we rein it in a way that's not like we're trying to, again, knock people down.
Right, but try to,
how do we like, if that isn't a good idea, can we it only becomes strengthened if you can refute the counterpoints.
Right. Well said. Another brilliant questions from Audrey Tran. No, I've enjoyed it a lot. Yeah, and I think listeners will enjoy it a great deal. Thank you, Audrey, for coming on.
Thank you for having me.
Yeah, it's good to see you.
Nice to see you too.
I'm back in plenary session, joined via Zoom by Dr. Karine Tawagi. Dr. Tawagi is a third-year hematology-oncology fellow at the Oschner Medical Center in New Orleans, Louisiana. She is the first of what I hope to be a theme here on this podcast of friends of the show and fellows joining us for a Journal Club episode. Dr. Tawagi, it's a pleasure to have you on.
Yeah, thank you for having me.
You're a friend of the show, is that fair to say?
It is. I was listening to the podcast starting in my first year of fellowship, and then you had a Friends of the Show session at ASCO.
That's right.
2 years ago in Chicago. And so we met then, and I've continued to listen to the podcast ever since. And it definitely has helped me with many of my own journal clubs in the last 2 years.
That's very nice of you to say. Thank you. We really appreciate your interest, and you're like the perfect person because, I hope that the podcast is resonant for fellows and that it's of some use for people going through these articles. You're here to talk about the I Am Brave 150 study, atezolizumab plus bevacizumab in unresectable hepatocellular carcinoma. And it joins the other IM studies, the IMbrave, Empower, Impassion. Power for lung, obviously, passion for breast cancer, and brave for hepatocellular. That's very obvious to me. Obviously. But you just did this in a journal club. Is that fair to say?
I did. I presented this for all the fellows at our own journal club here 2 weeks ago. So we have some interesting points of discussion, I think.
Okay, well, good. I look forward to it. I only read this morning. I was supposed to have read it a long time ago. And I didn't find as many things as I like to find, but I found a couple of points. So let's let you take it away. Why don't you, start by just maybe giving the listener some background about hepatocellular carcinoma and maybe this drug combination or why they did this study, maybe some of the background and methods.
Absolutely. So we know that historically hepatocellular carcinoma has very poor outcomes, especially when it's deemed unresectable, and multiple trials have tried to improve those outcomes. Often competing is the liver function of patients when they get to the stage where they need systemic therapy. So currently we had 2 category 1 agents that are approved in the frontline setting for unresectable hepatocellular carcinoma. We had sorafenib based on the New England Journal study in 2008, which had an OS of 10.7 months as compared to placebo with 7.9 months.
This is the famous Joseph Yové Sharp trial.
Yes, exactly.
Okay. All right. So that's one. Yeah.
And then a few years after that, we had the approval of lenvatinib based on a non-inferiority trial. So now that's also approved in the first line setting. And there's been a lot of other trials, phase 1 and 2, looking at immunotherapy, and atezolizumab and bevacizumab, which are studied in this phase 3 trial, have both been studied as monotherapy with response rate ranging in the 13 to 17% range.
I see.
And so this trial, the I Am Brave, is the first trial that has led to a new FDA approval as of May for this combination of this VEGF monoclonal antibody, bevacizumab, and then the PD-L1 inhibitor, atezolizumab.
Yeah. I guess it's interesting to me. I mean, that's a really great summary, I think, of the space, which is that, sorafenib came along, not a terrific drug, hazard ratio something like 0.7, survival from 8 to 11 months. But in the decade after that Joseph Yové paper, nothing beat it. I mean, we tried so many different other drugs, and then finally lenvatinib eked out a non-inferiority, which, I think I discussed on this podcast in one episode. I hate that trial because the margin is so big and the benefit was already so modest to begin with, so you really wonder if you're bumping up against placebo on the back end. But this is the first trial that comes along that supposedly surpasses sorafenib, so it is a noteworthy study, and it got approved. So that's where it falls. What about maybe tell us a little bit about who they studied and how they did it?
Absolutely. So the inclusion criteria for this study were patients over the age of 18 with locally advanced metastatic or unresectable hepatocellular carcinoma. They had to have had no prior systemic therapy and measurable disease as defined by RECIST 1.1. So biopsy was not mandated in this study. All of the patients were Child-Pugh A, and all had to have an ECOG of 0 or 1.
Nice.
And the exclusion criteria were obviously Child-Pugh B and C, co-infection with hepatitis B or C, as well as untreated or incompletely treated varices. So all of the patients actually had to have a screening EGD within 6 months of trial enrollment.
I see. And I guess I'll save your editorial comments for a little bit later, but I bet you have some thoughts on that. Okay, so I think that's what I took away from it. It's a classic frontline study focusing on Child-Pugh A, as we like to focus on the good old Child-Pugh A. I guess we can talk about that in a little bit, unless you want to talk about it now.
Well, I think you bring up a good point. Yeah. In terms of the current approvals, really we only have nivolumab that has been approved for the B7 patients, and this creates a clinical disconnect because oftentimes when we see these patients, they're not necessarily at ECOG of 0, and they're certainly not Child-Pugh A.
I spent some time trying to, one of the few points that I had was I tried to figure out, of 100 people that would walk into my clinic with de novo unresectable HCC, how many people are Child-Pugh A? And I went down this rabbit hole of looking at all these old publications that sought to validate Child-Pugh score. One of the things I kept seeing consistently was that people feel like Child-Pugh is actually not the best predictor of survival for HCC. It's a better cirrhotic survival.
Absolutely.
Yeah, and there are alternative scores like Barcelona and this click score that are better. But my best estimate was, at least 50% of people are Child-Pugh B or C. And probably that's even more because there's a referral bias in all these papers that healthier people are more likely to be seen. And so when you start talking about Child-Pugh A, and in this paper, I think they drew a distinction between A5 and A6, and you talked about B7, which is the better B. But when you start talking about A5, it's mostly A5. These are people whose liver function is pristine otherwise. Right. And I don't know how, I don't see that too often in the VA clinic that I was working at. I wonder how often you feel that you see it. Yeah.
No, I think polling the oncologist here when I presented this study, I think everyone, feels that they, for the majority, see Child-Pugh B, and oftentimes when the people are referred from the local regional
after they get beat up by all that local regional stuff.
Right. They're no longer candidates for systemic therapy, unfortunately.
Yeah. I think that's what I think many of us have found. And I think we'll talk about whether or not that has implications for the trial, but I think it does. Okay. Anything else we need to know in background, or should we jump to results? What do you think? You might have more stuff. Go ahead.
Yeah, so I just wanted to note, obviously, that the pharmaceutical company, Genentech La Roche, sponsored the trial, provided the trial drugs, collaborated with the steering committee on the collection, analysis, and interpretation of the data, including editorial assistance. And
my favorite thing.
I know your feelings about that, having listened to your podcast for some time.
Yeah, my favorite thing. Nothing better than having editorial assistance. I see it says, we thank blah, 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 blah from Blah Company, San Francisco for providing editorial assistance with earlier versions of the manuscript. Oh, thank you. Okay, good to note.
So the primary endpoints for this study were overall survival and progression-free survival, and then they had secondary endpoints including response rate, duration of response rate, time to deterioration of quality of life, physical functioning and role functioning, as well as safety and side effect profiles.
Yeah. And one thing I noticed that was interesting was the way they allocated the alpha. So their 0.05, they put 0.048 on the OS and 0.002 on the PFS. So like, in other words, even though those were both primary endpoints, they bet heavily that this was going to be OS positive and just a little bit of alpha for the PFS. But that doesn't change anything because it turned out to be positive by both metrics. Yeah.
Mm-hmm. Absolutely. So yeah, I think we can jump into results.
Okay, let's do it.
So there were 501 patients enrolled at 111 sites in 17 countries between March 15, 2018 and January 30th, 2019. The median age of patients was 64 to 66 years old, and 40% of patients were enrolled in Asia, excluding Japan.
I see.
In the supplemental report, the breakdown of race was 1/3 white patients, half Asian, and then there were only 2% patients that were Black. Mm-hmm. In terms of the performance status, 60% had an ECOG of 0 and the rest were ECOG of 1. Mm-hmm. As we mentioned, they were all Child-Pugh A5 and A6. Yeah. Some other interesting characteristics, 1/3 of patients had a clinical diagnosis of HCC, so I'm assuming those were the patients that did not require a biopsy. Mm-hmm. 2/3 of patients had extrahepatic spread, and half of patients had prior local therapy. And then in terms of PD-L1 expression, it was available for 36% of all patients. And in the combination group, 64% had positive expression, which they defined as TC or IC more than 1%.
I see. Okay. That's fair. I guess, it's one of those cancers where you don't really fault people for not biopsy proving it just because on imaging it's so characteristically HCC and you know when you see it, yeah. But I guess it's probably telling that the reason they only have PD-1 status on a fraction is that so many people are not getting biopsy proven.
Right.
Yeah, HCC. Right. Okay, that's good. What else do you got?
So I think we can jump to the endpoints.
Okay.
The combination group of atezolizumab and bevacizumab resulted in a 42% reduction in the risk of death compared with sorafenib, with a hazard ratio of 0.58 compared with sorafenib. And there was also a 41% reduction in the risk of disease progression or death with a hazard ratio of 0.59 as compared to sorafenib. Mm-hmm. Looking at the progression-free survival, the difference was 6.8 months in the combination group versus 4.3 months in the sorafenib group. Mm-hmm. And then in terms of the overall response rate, it was 27.3% in the combination group versus 11.9% in the sorafenib group. Mm-hmm. And I think that was a bit surprising for some people because historically sorafenib has had lower response rates than what is described in this study.
That's true. Yeah.
And then another interesting finding was that 5.5% of patients in the combination group had a complete response. And then in terms of the quality of life metrics, they had all the patients do a survey questionnaire on these quality of life metrics on day 1 of every cycle. And they found that the combination delayed the deterioration of patient-reported quality of life outcomes, physical functioning, and role functioning.
Yeah. And I think they used something like a 10-point decline on the EORTC scale as declining quality of life. But yeah, I think you're summing it up really accurately, which is it's a win for OS, it's a win for PFS, although quite modest, 6.8 versus 4 point something. Mm-hmm. And it's a win for quality of life. So it's a win-win-win. What's not to like?
Yes.
What's not to like? Yeah.
Well, we'll get into some criticism and limitations in a second. I also just wanted to note in the supplemental report, in terms of patients that went on to second-line therapy, in the combination group, 18.5% of patients went on to second-line therapy, and it looked like most of them got TKIs. And in the sorafenib group, 41.2% went on to second-line therapy, with majority of receiving immunotherapy, as we would expect.
Yeah.
And in terms of adverse events, any grade adverse event occurred in 99% of patients. About half of the patients in both trial arms had a grade 3 or 4 events. As expected, the most common grade 3 or 4 event was hypertension in both groups. And in the combination group, there were 15.5% of patients that had to discontinue the combination regimen due to side effects.
I see. Yeah.
And then I think of note, the grade 5 events in both groups were about 5%. So in the combination group, there were 15 patients that died. And then they list all of the reasons for the death. I don't think that, all the deaths necessarily can be directly correlated to the treatment. I mean, some of the causes of death listed were sepsis, cardiac arrest, respiratory distress.
Right.
So, but there was a good percent of GI hemorrhage, which obviously is of concern with the bevacizumab.
Yes. Okay. So there's some grade 5 AEs. They both, sorafenib's not known to be a walk in the park, but they both have sort of frequent rates of any grade hypertension, fatigue, proteinuria, as well as grade 4 and 3 events. Discontinuation rates were, I think, not terrible. But again, this is a highly select cohort, and everything we know about sorafenib is the discontinuation rates in the real world are really high.
Right.
Okay. And then
I think of note also dose modifications were permitted with sorafenib. So it was the starting dose was the 400 milligrams twice a day, but dose modifications were permitted. And then in the atezolizumab, bevacizumab, it was a standard dose of 1 day every 3 weeks for both drugs.
I see. Okay. And that's reasonable. Sorafenib has a terrible dose reduction schema that penalizes it. I've talked about on this podcast a couple of times, goes from 400 BID to 400 milligrams daily to 400 milligrams every other day, which are precipitous drops. And, when you could get in between that. So the first dose reduction is a 50% dose reduction. And that's why many of us have been critical in some of these trials. They start at the same dose, but then sorafenib has more steep dose reduction. So it's effectively on a lower effective dose. But, that's good to note. Okay. So what were the thoughts that came up in Journal Club? What do you guys think about the paper? What do you all think?
So I think, as we mentioned previously, obviously the fact that only child PUA with a performance status of 0 to 1, which are almost never the patients that we see walking into our clinics, does create the question of, can you use this off-label in the patients that don't exactly fit the trial parameters? Whether, we would get EGDs on all these patients. I think the consensus is most clinicians would get an EGD, especially if there are symptoms of portal hypertension to make sure that there aren't any varices, which might lead to an increased bleeding risk. I think that obviously we know that these drugs are very expensive. So I was looking at the cost of one dose of atezolizumab for the 1200 milligram dosing is about $10,000, and for a 60 kilogram patient to get the bevacizumab 15 milligrams per kilogram, it's about $6,000.
I see.
So one dose would be about $16,000. Again, sorafenib is also very expensive.
I know, unfortunately.
When I was looking this up, it was about $20,000 for a 1 month supply at the 400 milligram twice a day dose.
That's terrible.
But we know that globally, a lot of places are still using FOLFOX because really these drugs are unaffordable. Right.
Of the 170 countries they did it in, I wonder how many countries it's actually going to be affordable in. Right.
Right. Exactly. So I think that there definitely needs to be more studies looking at decompensated patients. However, it's hard to probably compare to sorafenib when that trial was only looking at the child PUA patients to start with.
Yeah, I think they had 2% of B in child PUB in that original Joseph Viove paper in Sharp. But yeah, it was almost exclusively, all the trials have been exclusively child's PUA, and that's been a real sort of, as you point out, a big question mark for generalizability. And we know doctors are going to generalize. The question is whether or not they're justified in those generalizations. Yeah.
Absolutely. And I think another interesting point of discussion that came up were this fear of anti-drug antibodies. So there was a paper in Clinical Cancer Research that described these anti-drug antibodies with all the immune checkpoint inhibitors. And atezolizumab was described as the one with the highest incidence of these anti-drug antibodies. But in some studies, it correlated to clinical efficacy, and in some it didn't, in bladder, non-small cell lung cancer, and breast cancer. So I think more research needs to be done in this area to identify whether this does come into play, but it does look like it had been most reported with atezolizumab as compared to the other.
That's interesting. I guess one irony is that in this example, we do have phase 3 trials for Nivo and Pembro. They both failed, and Atezo is the winner, but it's Atezo Bev. And I guess, one, question that comes is, obviously, is the Bev needed? Is Atezo going to win on its own, or does it need the Bev? And I guess we'll probably never know the answer to that question. And so they'll likely be given, just as protocol was, to give them both together. Mm-hmm. Any other, I found a couple things I'll tell you about in a second, but any other things you want to mention?
I mean, I think, just the breakdown of patients enrolled in terms of race.
Oh, yeah. What did you find? I didn't look at that. Is it mostly Caucasian?
2% Black, and then a third Caucasian, half Asian.
Oh, yes,
I think historically, some of the patients that have the viral induced liver disease tend to have better responses than the North American population that might have non-viral induced cirrhosis.
That's a good point, because I guess it looked like it was split between Hep A, Hep B, and non-hepatitis HCC, like alcohol use typically. And it looked like in the forest plot somewhere I read it, and maybe not the paper, but the supplement, that overall survival was clearly beneficial in viral-related hepatocellular carcinoma, but it wasn't so clear for the non-viral HCC. It looked,
right. No, you're right. Yeah, it was Hep B, Hep C, and non-viral.
And so your point is that, by going to Asia, of course you're going to enrich. Of course you're going to enrich in Hep B. But the question is, in the U.S. population, where you have more and more liver cancer as a sequela of perhaps non-viral processes, are you going to get the same benefit? And there's a question mark, I think, whether or not this combination works in that.
Oh, absolutely.
One thing I found that I thought was interesting was it's an open label study, so it wasn't double blind. Yes. And so they didn't give placebo infusions, so everybody knows what the patients are getting. And I can imagine you're very happy if you get the new shiny Atezo Bev, and you might be annoyed if you get the old-fashioned sorafenib. Right. And then I noticed that the duration of treatment was 7 months for Atezo Bev and 2.7 months for sorafenib. So I said, okay, 2 months for sorafenib. I definitely know that the real world utilization of sorafenib has a very short duration of treatment because people don't like that medicine. But this is a clinical trial enriching for Child-Pugh A and mostly A5, ECOG 0, clinical trial where if you read the, the inclusion criteria, they also say like, you can't even have hypertension to 150. There are all these kind of unique anti-angiogenesis kind of IC included. So it struck out to me that I'm like, okay, this is a really selected cohort. Duration of treatment is 2.9 months. So I was like, let me check the other trials. So I pulled the lenvatinib non-inferiority study. And in that lenvatinib study, the duration of treatment for lenvatinib was 5.7 months, but it was 3.7 months for sorafenib. So it was longer for sorafenib than in IMbrave 150 by a month. And then I pulled the original SHARP study from 2008, and I said, what is the duration of treatment? The median duration of treatment was, and then one thing I should add is, IMbrave, you're allowed to give sorafenib post-progression. That was an option to investigators. The duration of treatment in the 2008 paper, a decade earlier, was 5.3 months in sorafenib, compared to 4 months in placebo. So how is this paper, the duration of treatment is less for sorafenib in this study than it's less than half of what it was 10 years ago, and less than placebo. And I guess my question, though I cannot prove it, my question is someone taking a dive on the control arm is my question. Like, if you know that this, in the original SHARP study, the company that comes to you is saying, we want to get sorafenib approved, push the dose of the drug, keep the dose high, keep it going post-progression, let's give it the best shot at improving OS, and we win the OS benefit. And in this trial, a decade later, they come to you and they say, we've got this new immunotherapy combination. Do what you want with sorafenib. Their doctors are giving it for half the duration, even though the patients are probably as healthy or probably healthier than they were a decade ago. And people always say that, well, the sorafenib arm, they lived, median survival was like 13 months, and it was 11 months a decade ago. But if they live 13 months with 2 months of sorafenib, if you gave them 5 months of sorafenib, maybe they live 17, 18 months, and some of that benefit would be eroded. That was just a thought I had. I mean, it's obviously you can't prove it, but it's one of the things that a placebo control would have helped, because doctors would not, a double placebo control might have made doctors more reluctant to discontinue things a little bit prematurely, push post-progression.
Right. No, that's absolutely true. And, I know they did mention in the article that they designed it this way to spare patients from 2 placebo infusions.
Yes. And
that as a result, they had blinded review of the imaging to minimize bias. But I think you definitely bring up some good points that.
Yeah, I want to thank so-and-so of such and such corporation, San Francisco, for that clever play on words to spare them the infusions. Yeah. So, I mean, it does do that. It spares them placebo infusion, but it also makes it abundantly clear who's getting the drug. And so if there is a psychological bias against sorafenib, which there well could be, and I guess I would say that it would be one thing if the duration of treatment was 5.5 months, but 2.7 months. Every time there's a new trial, the duration of treatment of sorafenib gets shorter and shorter. It was 5 months, then it went to 3.7 months, and lenvatinib was non-inferior. Now it's 2 months, and this is superior. In the future, it's going to be half a month, and the drug will be a game changer. If you don't give sorafenib at all. So I thought that was interesting. But other than that, I mean, I think you and I have come to the same conclusions. How generalizable is this? How, it's clearly maybe, you might lean that way for the fittest of the fit people, but then again, you probably dog the control arm a little bit. Is it really, the real sad part here is that this kind of approval is probably not going to transform outcomes for this disease, because the reality is most people with this disease also have hepatic dysfunction, and we don't have really great drugs for hepatic dysfunction.
Right. And I think that, in the future, trying to catch those patients that are still getting liver-directed therapy sooner, and there's a lot of trials ongoing looking at, local therapy with systemic therapy concurrently or sequentially. I think there'll be a lot more studies coming out in those areas to see if maybe that does improve outcomes. Right.
So overall, what was your takeaway from the conference? What did people say? They're going to use it selectively, is my guess, but what are their impressions?
I think impressions overall, people are going to use it up to a B7, even though that would be considered off-label in theory.
Boy. Okay.
Okay. Most people would get a screening EGD if there are any signs of portal hypertension. And in terms of what to use second line, if you do use this in the first line setting, obviously a TKI, but the question was as to which one would you use? Yeah,
I know. Now you have no data for that decision. Regorafenib, Cabo. I mean, none of these got this in the front line. Okay, I guess I would say, would I use it in an A5 patient? Yeah. A6? Question mark. B7? I wouldn't. That's just my gut. And then I would say that if you did a trial where people push the sorafenib hard, the benefit would get smaller. Would it evaporate? Question mark is my thought. But I guess one question that will be is if any of the responses are durable. That might change people's tune. Is if we find that in 3 years, 5-year follow-up, there's some fraction that is, the tail on the curve settles somewhere. I know in these immunotherapy drugs, we're always hoping, but half the time we're disappointed.
Right. And I mean, there were 5.5% that had a complete response in the combination arm. So
we'll see where they are in the future. What
happens with those patients.
Yeah, it's been definitely sort of a mix across tumor types. But yeah, I think that's a great summary of the study. And a very interesting study. Liver cancer trials, they all suffer from, we're only really studying the tip of the iceberg of people with disease that come to our clinics. And so, it would be great someday to just have trials dedicated in Child's Pew B, for instance, to try to figure out, what's actually the best strategy.
No, absolutely. I just wanted to also mention that there are 4 other phase 3 trials that are ongoing looking at immunotherapy in unresectable HCC. So I think that, this regimen, we'll see, it might be also
eclipsed,
fighting with other regimens. So there's the LEAP 002 trial, lenvatinib and pembro versus lenvatinib. the Cosmic 312 trial, cabozantinib with atezolizumab versus sorafenib.
Oh boy.
There's also the Checkmate 90W, which is nivo-ipi versus sorafenib or nivatinib.
Okay.
And then finally, the Himalaya study, which is durvalumab and tremelimumab versus durvalumab alone versus sorafenib.
Oh, wow. Good. So everyone wants to beat on sorafenib. Well, good news is one of those studies will be positive by chance alone. But I bet that the duration of treatment of sorafenib is going to be like 2.1 months. We got to go down for those studies. It's got to be less. But you're right. Maybe one of those will eke out a win, and then we'll have to think about it. And then it'll be, I wonder which is the most costly. My guess is probably the most costly is the one with the cabo in it. What was that? Atezo cabo?
Yeah, atezo cabo versus sorafenib.
Yeah, that's probably the most costly.
Cosmic 312.
Cosmic.
So we'll
I like that title. Cosmic. All right, so that's good. There's a lot of interest in this space. There's been no shortage of randomized trials in HCC, a very tough disease. All right, that's terrific. So yeah, thanks for doing this. I thought it was a very interesting discussion. It was a great presentation. Thank you, Dr. Tawagi. Thank you for coming on. It was a really great presentation, very interesting discussion. And thanks for doing the first Visiting Fellow Journal Club on plenary session.
Oh, I'm very honored to have been the first visiting fellow. So I hope to see more of these kind of podcasts in the future. Yeah.
And you should put it on your CV so that someday it says visiting fellow. I hope people take it seriously, but you never know. But yeah, thanks so much for doing this. Yeah, hopefully there's a couple people who emailed me some other trials. So we hopefully have a few segments coming out, but thank you.
Yeah, that would be great. Hopefully we'll see if we meet at ASCO again next year.
I know, if it's in person. Yeah.
Yeah.
You've been listening to Season 3 of Plenary Session. Plenary Session is produced by Kiana Klossner. Music by Ian Straley and Audrey Tran. The views expressed on Plenary Session are those of whoever said it and no one else. Plenary Session is not medical advice. Follow us on Twitter at plenary_session. Until next time.